This is a continuing discussion on the findings we found on Reed's cases using Triton as an irrigant, along with using a photona laser and a sweeps method. When you look at these cases, we found these cellular-like structures that we weren't sure what they were. I thought maybe they were mesenchymal stem cells because it seemed to be interconnected with each other and right against the mineralization front. But the question is, well, if they're not cells, what are they? And when we looked carefully at the cells in many of the different specimens, we found that they were all pretty much the same size, about 5 microns in diameter or a little smaller, and that they seem to appear in sheets or layers just as cells would. And they're only in Triton treated cases, and they seem to be all together. When I posted these findings on the IAE site, Craig Rhodes came up and said, well, I wonder if these are undergoing discocyte echinocyte transformation. And I didn't know what that was. I had never heard of it. He referred to them as crenated red blood cells. So we looked it up, and sure enough, crenation is a term used to describe an object having a scalloped or rounded tooth edge to it. And this certainly matches the structures that we were seeing. And then I went in and did a search, and we found out even back in the 1970s, this phenomenon was well known, and they changed the terminology a little bit in calling the normal erythrocyte as a discocyte for the biconcave disc, the usual circulating form of the red blood cell, and echinocyte, after the Greek echinos, meaning sea urchin, for the crenated spiculed form produced by alterations in the intra or extracellular environment. So this biconcave disc form of the discocyte, it's dependent on a delicate equilibrium of multiple forces, both internal and external, that is poorly understood. As this transformation takes place, the biconcave disc changes its appearance into this crenated form, or the echinocyte form, that looks almost like a white blood cell. So the summary of this is erythrocytes exhibit disc sphere transformation. The transformation can be reversible or irreversible. They're referred to as crenated spheres. The discocyte refers to the biconcave disc. The echinocyte refers to the crenated sphere. And there's an equilibrium between these two forms, and it's controlled by things having to do with the lipid bilayer in the outer cell membrane, which becomes weakened or lengthened because of osmotic or other chemical changes in the environment. So this makes a lot more sense than thinking of them as mesenchymal stem cells or a non-cellular form. They are definitely cellular, and these are definitely red blood cells that have been changed somehow by the Triton. And when you go back and look at the SEMs with this knowledge, now you can see how these discocytes actually transition over to echinocytes, and you can see the process in all the different stages of transformation, where you had these early projections on the erythrocyte surface, and then as the transition continues, it turns into an echinocyte. So now these pictures are starting to make quite a bit more sense. Here's another one from Reed's case, and you can kind of see one stage where the RBC is starting to transition, then another where it's further along the transition, and then the final size. And then when you go in and look at some later papers that discuss the transition process even better, you can see how this process starts on the surface with these little projections and progresses quite significantly as the red blood cell changes its shape and that outer membrane becomes crenated. So now these SEM pictures are making a lot more sense as you look at the entire process through Reed's cases, and why we thought these were stem cells, because of the interconnections between the red blood cells, which typically you don't see in the normal red blood cell. Now, another paper has this about ethylene glycol, which is actually in Triton, and how that accentuates the transition to echinocytes. It's a function of the ethylene glycol. And so Triton has ethylene glycol in it. And when you read this paper, now it makes even more sense. So when you have the activation by the laser of the ethylene glycol, it affects the outer membrane very, very quickly. And that's why there are so many of these echinocytes in Reed's cases where sweeps was used with the Triton. So now it's making even more sense for why we are seeing this only in Triton cases. And we see these projections, and it turns out in the work that was done is these projections actually contain the ethylene glycol. It sequesters in these outer projections. And there's a picture from one of Reed's cases where you can actually see the concentration of polyethylene glycol in that projection. So now the pictures make even more sense as we're looking at these. And as you look through the stitched images, now you'll be able to actually make some sense of what these figures actually represent. So I thought you'd all be interested in kind of seeing what some of the research that's been done on this that I was unaware of. But thanks to Craig Rhodes for pointing this out. It really pointed us in the right direction. And I hope you all find it very interesting.